So let me start off. This is Udit Singh, application scientist from Bremas Life Sciences. And my current session, we would be discussing why NGS. What edge does it deliver to the user? That basically what benefit it offers um, to your research. So that those capabilities we are going to discuss. Uh, then we are going to understand how the workflow goes around your Illumina NGS. So from your sample to your final result, what is the intermediate process that is involved in this entire NGS setup? We'll discuss that. We'll see. I have some video also. Uh, then I'll be showcasing the entire Illumina sequencing range of sequencers. So basically, it's scalable. Uh, it is for every user, a novice or a production scale, whichever the user falls in. So apart from that, we'll discuss the library prep options and the certain data analysis options that Illumina has. So let's start off. So Illumina, with its highly accurate and deep sequencing, and its sequencing by synthesis method is very highly accurate. It offers you most of the data in the Q30 range. So with that, it gives you that NGS experiment will give you the qualitative information as well as your quantitative information. So as Ashutosh discussed, you can check for in the clinical applications, you can check for your germline mutation or somatic mutation. It could be heritable disorders, hereditary cancers, hereditary disorders. An individual can be a carrier also. So prior to marriage, you can check your carrier screening. So you can check the mutations. You can see pairwise differences, whether there is a difference in the genetic makeup of the two samples. So apart from this, if you are working on genetic engineering and let's say you have made some modifications and you want to check whether it has turned out to be okay. So that can also be checked with this NGS. So basically, you will run your samples and you will see whether the modifications that you have done have they occurred successfully. So these were the qualitative informations that you can get, and also because the method is highly sensitive, you can have your quantitative informations too. So you can check for aneuploidies. You can so basically this your NIPT. These are count based methods. The NIPT and PGS that was previously discussed. So this can be also discovered. You can check for expression changes. You can check your gene regulations. You can basically sequence the methylome also. So epigenetics. This can be done, and also your environmental DNA. You can profile what microbes are present in your sample or whatever environmental DNA you are targeting. So these are the information that you can get from NGS, and that is the edge that it offers. It is a very deep sequencing. It gives you a broader discovery power, and also it's highly sensitive. So you can rely on this. So now, with the onset of COVID, there was a requirement of a test that is highly accurate and also offers a high throughput capability. So Illumina came out with its COVID-seq test and assay. So basically, this test actually covers your entire COVID genome. Uh, it can be it can be used for as a test as well as for surveillance. So it covers your entire COVID genome, and also the throughput is pretty high. So this is 3072 samples in one go. And also it's foolproof because it has some process controls involved. It has 11 human gene targets that are built, and also there is a built pipeline, Dragon analysis pipeline, that will give you your result. So this was the test developed by Illumina. When this, so it was first done in IGIB in Delhi itself. So the yeah, it offers you high accuracy and high throughput. Uh, let's move on to let's understand what is the workflow, the NGS workflow. So it starts off from your sample. So your sample type can be anything. Uh, it can be from any source. Uh, so basically, it should have your nucleic acids, and the first step would be your sample prep. So from your sample, whichever source, whichever type, you will extract your nucleic acids. You will have your DNA, RNA, and then you will go on for your library preparation. Now, let's understand what is library prep. So basically, library prep, in simple words, is your making your nucleic acids compatible to your sequencing. The samples, the nucleic acids cannot go directly onto the sequencing platform. It needs to be modified. So basically, because this is a short read sequencing, the nucleic acids need to be fragmented, and they need to come to a certain range. After that, certain modifications, certain adapters are added to it, and then that is basically your library, and it is compatible to your sequencing platform. Uh, Also, the any modifications that you do this any nucleic acid, it turns out the library is same for all the applications. The end library will be same for all the applications. So also it is called a library because you basically fragment it, and then there are many short fragments representing the whole target or the genome that you are trying to sequence. So that's why it's also called a library. Then when you are ready with your library, it goes on to the sequencing, and the sequencer will basically do your SBS, sequencing by synthesis process, and generate data. It will basically read each nucleotide individually in each fragment, and then you will have your data that can be analyzed as per your need. So this is the common workflow. Now let's see the library, what the library fragment looks like. So basically, this is a library fragment. Any application or any source of sample, anything, the end library fragment will look like this in every case. So basically, this gray region is your DNA insert. That is the DNA fragment. And then this is your basically the modifications that we have done. So it has your three basic parts: P5, then your index, and then your read one sequencing primer. So this these three regions are involved in your sequencing. So you will have a bit more clarity in the next when I play the video. So basically, you will understand what are the individual uses. So P5 basically is involved in your cluster generation process. So it will hybridize to the oligos present on the flow cell. Indexes are used for multiplexing. So basically, all the samples will be pooled together and they'll be put into one single run. So basically, if you want to segregate, you need indexes. So these are unique sequences that are introduced during your library preparation step for each sample. And at the end, when the sequencing is completed, with the use of indexes, it is segregated. And read one sequencing primer binding site is basically where your primer binds and then your sequencing initiates. So let me just play this movie.
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
Hi. The video might have given you some clarity over the sequencing by synthesis process. So basically, you have your library fragment. It is then clonally amplified. So how it is done? It is done through a bridge amplification process, and it generates clusters. It makes clusters. So onto the flow cell. So this is a flow cell. It has your oligos bound to it. Yeah, this library fragment is introduced. It goes and hybridizes to these oligos, and then bridge amplification happens. And so basically, it generates a cluster. And cluster generation is basically done to increase the signal, because the camera just cannot detect a single nucleotide incorporation. It needs something more. So it needs a high intensity. So that's why clusters are created. A clonal cluster is created so that when the incorporation happens, the signal from that cluster will be detectable by the camera. So this is the cluster, and then sequencing happens. So your individual nucleotides go ahead, bind the camera, the LED excites, the camera will image, and then you will have your base call. So basically, it is sequencing by synthesis. It sequences one nucleotide at a time, and so for like some cases like homopolymer regions, it basically has no effect in your SBS. So apart from other sequencers like ion torrent, which is affected by the homopolymer range, Illumina is not, due to this SBS chemistry. So it basically maps one location. So this single point is your cluster, and it will register the signals coming from that point uh, at every cycle, and it will see uh, what bases are incorporated, and then the complementary is basically your original. So this is how it does. And then it will do your so this is how it registers, and you will have your data. So how can the samples be mixed? So during the library prep step, as I already discussed, we incorporate index. So index are unique sequences, and it is added to the sample during the library prep step, and it basically gives it a unique identity for it to be segregated at the end, at the data analysis step. So these so dual indexes are placed on each sample basically, and with this, so this is the entire data sequence. With the help of the indexes, the it will be segregated and demultiplied. So this is how it basically goes in, and it is then demultiplexed during your data analysis step, and then you will have your individual data. So from sample one, whatever data you have and individual samples, you will get the data. Then when you have your individual demultiplex data, you can go for your further analysis part using data analysis software, or you can utilize your base space. So base space is a analysis software or from Illumina, and then you can have your variants. It could be anything for your VCF file, so that can be done from this. Now, when you plan an experiment, basically there are certain questions that you have to ask yourself. How much data do you require? First thing. Then, what would be the read length? And according to that read length, whether do you want it to be a single end or a paired end? So data is basically whichever genome you are targeting, or whatever region you are targeting, and how much coverage do you need, how many times do you want to sequence. So then you will have your the total data that you want. Also, whichever genome, the size of it, according to that, you will see how what would be the read length. And similarly, along with this, you will decide your single or paired end read. So whether if it is a very small, so let's start off with your, let's see this. Wait, hold on. single and paired end. So why single or paired end? So basically, if you your data requirement is very less and your target is also small, in case of let's say small RNA, so basically it's just 22 nucleotides. So you can basically cover it in a single uh, read itself. If you are just doing a basic counting experiment, and when you are doing something like a de novo or something which has in which you need more data and you want to align it, so basically then you go for your paired end sequencing. So paired end sequencing basically offers you two benefits. At the end of sequencing, there is a loss in data quality. So if you are overlapping it, then you might cover it in your second read. Second is the alignment part. So basically two sequences needs to be aligned to the reference genome. So basically you have 2 points to align. So uh, it goes for it gives you a better alignment in that part. So this is the benefit of paired end sequencing, and Illumina has that. So Illumina offers your paired end sequencing, and also in you can, if you have de novo, if you want to do de novo sequencing, you can go for the larger reads. That 2 cost 300 that is offered on Illumina MySeq instrument. So, so we have I have given you how the what edge does NGS Illumina NGS offers, and also uh, what is the workflow. Now let's understand what are the sequencing systems that Illumina has to offer. So basically, we have this range. It starts off from your iSeq, the smallest machine, iSeq 100, to our biggest machine, NovaSeq. So this one generates around 1 GB of data, and this one generates around 6,000 GBs of data. And intermediate, we have certain machines, the benchtop ones. This is also benchtop. These MiniSeq, MySeq, NextSeq, and then you have certain. So this one will overlap in your this NextSeq 550 has flexible power, so that's why it comes in low throughput as well as your high throughput. And then we have NextSeq 2000. It is the recent machine. It's highly advanced with no fluidics inside. Also, this one has no fluidics inside. So this is our entire range. Let's move on to individual sequencers and let's see what it has to offer. So our smallest machine, iSeq 100, it's on a, single channel chemistry. It basically has only one channel, and it's basically on and off. So 0101, that's how it base calls. And with this, it offers you 1.2 GB of data and 4 million reads. And so this machine is pretty simple. It has no fluidics built in, so low maintenance, no washing steps. So and also this is also user installable. So what you can do, so basically the data is less, so you can go for small gene panels, small targets, amplicons. So this is what you can do in this sequencing system. Uh, so the main benefits, main highlights are this is a very low cost machine, highly reliable, because it has no fluidics inside. It basically requires low maintenance, and all the fluidics and all the liquid part is basically built into the cartridge. All the fluidics part is built into the cartridge, so that's why this machine has basically less moving parts, and so that this is more reliable, simplified workflow. Because after the library prep, there are certain steps that needs to be done prior to adding it to the sequencer. There are just simple pipetting steps, but here that part is also much more simplified. So no denaturation of the library is required. Onboard denaturation happens in this machine itself. Setup is pretty simple. This is a touchscreen LED screen, and with our user-friendly interface, you can just set up the run within a few minutes. And this machine is also, because very simple, it is customer installable. So basically, you just receive the box, you uh, place it onto your benchtop and plug and play. So that's how it goes. Uh, Coming on to um, this mini seek. So it's basically offers you higher data than your iSeek machine. Um, this offers you 7.5 gigabases of data. So 25 million reads. So this mini seek machine offers you two options of sequencing: mid output and high output. So two simple options. And basically it has less data. So the applications here are also your small genomes, and basically this mini-seq machine is mostly used for in India, it's mostly used for your HLA typing applications. So the major highlights are low capital. So this is again from a starting range. So this is a low capital machine. Simple onboard data analysis, so LRM built in. We'll discuss LRM in further slides. And also you can go for your small genomes. So small RNA sequencing can be done, resequencing can be done. So resequencing is an application wherein when you want to do some if you want to refer, so you have the reference already there and you want to do it. So this can be done. Also, these machines are also utilized for your, well, let's say you want to set up a run in our biggest sequencers, the NovaSeq or the NextSeq 2000, let's say. So the run cost is pretty high, and we want to validate those libraries, whether the performance of those libraries are good to be sequenced on the bigger machines. So that is also done. The validation parts are done in our lower range of sequencers. And also you can go for your if you have certain amplicons you want to sequence, and small enrichment panels can also be done in this machine. Now coming on to our MySeq. So here are certain, this machine will has certain highlights. So basic certain highlights. So this actually offers you paired and 300. So this is the highest read length that is offered by MySeq in the entire Illumina range. So PRN 300 is offered by MySeq. So, and the data is 15 GBs, and 25 other specs are mostly similar to your mini-seq. This is the major highlight that is offered by MySeq, PRN 300. And and also another big highlight of this machine is that it has a very high scalable sequencing option. So basically you have four types of kits available: the nano, micro, a standard version 2, and a standard version 3. So MySeq is the only machine that offers your PRN 300, the longest read length, as well as very high scalable options of sequencing. So if you have less data, you can go for nano. If you want more data, you can go for V3, or also if you want to go for the 600 cycles. So 600 cycles are basically the paired, and 300 is offered in the version 3 only. So these are the applications. So again, mostly the specs are similar to mini-seq, so small genomes can be done. And apart from this, one major highlight, de novo sequencing, wherein you will require a larger read length. So that can be done. Also library QC. So library QC is that part which I discussed. So if you want to QC check the libraries, check the performance of the libraries, so this can be done on the smaller machines prior to going to the bigger machines. So the basic, so you have your long read and you have your scalable options. And again, the workflow is pretty simple, and it's simple for all the Illumina sequencers. So now let's move on to NextSeq 550. So now this is basically a big upgrade here. So from 15 GBs, you move on to 120 GBs, and the reads here are 400 million reads. Now, so this is where you will unlock certain applications that could not be done on the sequencers that we have discussed previously. So 120 GBs unlocks some applications like your transcriptome, like your exome sequencing. And also, if you want to go for a human whole genome, that can also be done, but only one sample can be done. But yes, it offers you the capability of doing that. So two options, high output and mid output, two simple options you have. Uh, so the higher one, the high output one offers you the highest range. So this is 120 GB and 400 million reads. And apart from this, you have your hold on. So this gives you a high throughput applications, and you can basically go for your transcriptome, wherein you will, if you want to do a human transcriptome, you will be targeting 50 million reads. So 50 million reads basically was in its the NextSeq 550, from where you can start your transcriptome experiment. And the previous sequencers, the highest reads were 50 million reads paired, and so basically transcriptome is not possible there. So NextSeq 550 is the machine from wherein, if you want to do transcriptome, this is the lowest machine that you need to have. Also, you can go for exome panels, and if you want to do a whole human genome, that can also be done. So also, this machine has a major highlight that it can also go for your array application. So so basically the Infinium arrays, they can, this machine can scan those arrays, so you can also do that part. uh, Coming on to our next upgrade, that is your NextSeq 2000. So now here, with the NextSeq 2000, Illumina has made major advancement in this machine. And so basically this machine is fluidics friendly, fluidics free, sorry. It's fluidics free, and so basically less maintenance. It does not you need not perform any wash. You So all the fluidics are basically built onto the cartridge itself. And it has also modified, upgraded its imaging modules and the camera, so it's higher resolution. And basically this led to your smaller flow cell. So this is a very high density flow cell, and basically it's much more smaller than the NextSeq 550 flow cell. And apparently this offers you much, so three times more data, 360 GBs of data in this, the biggest flow cell that this offers, and 1.2 billion reads uh, with the NextSeq 2000 P3 flow cell. So this machine is the recent one from Illumina. It is fluidics free. It has a higher resolution, and also it goes for your miniaturized sequencing reaction. So basically, the cartridges, they are built so the technology is such that they require less reagent. So less reagent means the cartridge will be also small, and small volumes means your storage footprint will be less. So basically, you will be saving on your storage cost also. So that is the edge that this machine offers. Also, uh, NextSeq 2000 has your Dragon Bio IT platform built in, and also this machine, uh, unlike other Illumina platforms, it's based on Linux. So there's also the user interface is pretty simple. The GUI is very simple. So when it boots up, this is a screen that you have. Uh, it's pretty simple. The workflow steps are very easy, and so basically onboard denaturation happens inside this machine itself, and the run setup options are also diverse. So basically you have many, you have your local, you have your cloud, as well as your hybrid options to set up a run. So let's move on to our NovaSeq 6000 system. So this is the biggest sequencer that Illumina offers, and it is not a benchtop system, it's a standalone. So there's any sequencing, any project, you name it, and it can be done on NovaSeq. So basically, it offers you 6,000 GB of data in its full capacity. And also, let's discuss in the next slide. So basically, these are the options that it offers. So from SP to S4. So this is the flexibility and the scalability that this offers. And the flow cells are basically patterned, very high density patterned flow cells. So a S4 flow cell offers you 3 TB, and two of these flow cells can go in a single when you run. So basically, NovaSeq has two. It can hold two flow cells, and they can be run uh, independently. So when you go for two of these S4 runs, it offers you 6 TB of data. Uh, let's move on to understand what can be done. This will give you a glimpse of how many samples you can basically do. So basically, this is a high throughput sequencer. A human genome on a single S4 flow cell, you can do 24 samples. If you are going for and also if you are targeting 30x genome coverage. Uh, when you are doing exome, let's say exome, and if you are targeting 100x genome coverage, 250 of these libraries can be sequenced on a single S4 flow cell. And transcriptome, when you are targeting somewhat or more than 50 million reads, so basically then you can go for your uh, 200 samples. So this is a high throughput sequencer, and also individual lanes can be utilized for individuals library type, also different library types. Basically, you have to follow a different workflow for that, but yes, it offers you that capability also, that the individual lanes can run different libraries. So this was all for the entire range. So basically, you have your uh, from iSeq to your NovaSeq. It basically enables each user. So a user can be new to sequencing. They can basically go for your smaller sequencers, the iSeq, MiniSeq, MySeq, and there is a sequencer NextSeq 550 which has flexible power. It can go for a new user can also use it, and also an experienced one, or the one which has higher data requirements. And also then we have your the production scale, your NextSeq 2000 and your NovaSeq. So all this, the Illumina has an entire range. It enables each of all the users, whatever their data requirement is, they can go for whatever sequencer they seem fit. Now let's move on to the library prep options that Illumina offers. So uh, here are certain applications, that the major applications. So you have your DNA. DNA libraries can be made, and the RNA to check your gene expressions. Then also you have your methylome study, wherein you can check for your gene regulations. So these are different library prep methods, and Illumina offers different library prep solutions for each, catering to each application. So these are certain kits are listed here. So some of them are can be clinical also. Some can be a normal, like a whole genome library kit will be just for sequencing your DNA. So basically, you can have DNA from any source, any sample. So that can be prepared from that library prep kit, and then you can go ahead and sequence as per on the sequencer of your choice, whatever data you want. So there are certain kits that are very broad range, and there are certain kits that are very specific, like your clinical library kits. Let's say your very CPGS. So they are very specific for certain applications. So, and also these library prep are automated on your the major players of automations out there. So Agilent, your Hamilton, Beckman, Perkin-Elmer, Deacon. So so basically these protocols can be automated or are automated on these automated automation platforms. So if you want to go for high throughput library preparation, that can be also done. So there are third party automation this machines available. So we have understood the library prep kit part. We have understood the sequencing, and we have understood the workflow. Let's understand at the end, when you have the data, uh, what solution does it actually offer? So
That we are not covering informatics here. That's been told very clearly.
Okay.
Right. So let's skip this part.
Mm-hmm.
Yes,
Okay. So
because they
basically,
have
it is
two
just
days.
okay.
Yeah. So let's take the Q&A. You have anything else apart from informatics? Then you can go ahead.
No, no, no. So basically, that was just LRM, nothing more.
Yeah. Yeah. Then let's hold this. We can take the
Okay.
questions. Yeah.
Yeah, sure, sure. So that was all for the session. So let me just summarize. In the session, I have covered your basic uh, NGS edge that it delivers, and also the workflow. So from the sample, how it's the library is made, and then how it goes onto the sequencer and how it is sequenced. Uh, I have showcased the entire Illumina range, so from your smallest sequencer to the biggest sequencer, and also the library prep solutions that Illumina offers. So that was all for the session. If you have any questions, you can ask them.
Yes, there is a question in chat. It's from Saga. He's asking what strategy should we go for miRNA-seq?
Okay. So it's basically miRNA, very small. Uh, so it's a very small RNA, and you can basically go for your small RNA kit, and that can be done on your these normal small sequencers. It can be done. So there's a kit, 2-seq small RNA kit, that can be utilized for your library preparation, and whatever data you require according to that, and then a number of samples also, the number of samples and the amount of data you want. Accordingly, your run can be set up, and basically here you target for a very small read length. It is single end. So for your miRNA, you will go for single end because it's a very small size.
Yes, the next question is which one we can use for ChIP-seq? I think they're asking the kit name, I guess. Am I right, Priya?
Okay.
Hello?
Kit name and instrument. Okay.
Yes.
Okay. So this, I think I will have to get back. I will just get back over the email which kit for ChIP-seq can be utilized. And instrument is basically so it depends on how many samples you have and the data you require. So that in that way you decide which sequencer to go for. So this has to be decided by the user. According to that, we can suggest you which sequencer you can go ahead and sequence this. So regarding the library prep kit, I will get back to you. I will write it over mail. So I will share it with you.
Yes, I think the next question is on me. What are all the information we can extract out of RNA-seq? So we can get a splice site variance, we can get an expression profile, and we can get a differential expression profile. And downstream, of course, pathways can be analyzed. Gene ontology studies. So basically, what question we are asking, that it depends upon that we'll be doing the downstream analysis. So pathway is one of it. There are multiple ways it can be analyzed. I think Pine Biotech have a plan for you. I think they will be using some tools specifically on this, I believe. So you have seen
Yes,
the
I
answer.
actually just wanted to interrupt here for a second only. So at the informatics part, we have 2 day sessions, like over 10 years of training, which is planned for tomorrow and day after tomorrow. So anyone who is like inquisitive about the informatics part, we would like to invite them for tomorrow's session. Thank you. Please. Yes, Udit, you can go ahead. I was just telling about the informatics session that you were explaining that tomorrow we have with Pine Biotech.
Yeah.
So tomorrow and day after tomorrow, we have 2 days of genomics, metagenomics, and transcriptomics data analysis, and then that will be followed by some of the chemoinformatics strategies that can be utilized for drug discovery. Thank you.
Thank you, Mohit.
So yeah,
Udit,
yeah. Yeah, Raja.
I think there's no more question. Thank you so much for the talk, Udit, and thank you for all the responses, like say for all the replies for the questions. I think one more is coming. Can base space be used for analysis RNA-seq? So Saga, I think I can take this offline because we are not discussing any Illumina related solutions here, right? So anything on the instrument sequencing related, we can answer. Analysis, since it's been covered next 2 days, we are not taking any questions on that. But offline, if you want, you can reach Premas at any time, right? Yeah, thank you so much. So thank you so much again, Udit, for the talk as well as for all the responses. Let's bring Nitya in picture now.
Yeah, thanks, Raja. Let me just stop sharing my screen.
So Nitya, are you in?
Hello. Yeah, Raja.
Yes. So Nitya is our product manager for 10x. He'll be discussing about all the 10x related solutions now. Can you share your screen, Nitya?
So is there any break or we can go ahead? Because I was thinking that there is a break. The timing is 1 p.m. to 2 p.m.
Are we having a break, Mohit? Or we can continue? What do you say?
Yeah, we can continue. So this is the last session, I think, right?
Yes. Yeah, this is the last question. Yes.
Yeah, so let's conclude, and I would want everyone to be free. Is it like holidays and all, right?
Okay. So we don't want to break the curiosity. Yes. Yes.
Yes, it is going really well. It's wonderful sessions, and I think it's all connected. So we are all inquisitive to learn more. Thank you so much.